Good morning. So I guess no one joined, only Humera. Good morning, Humera. I'm gonna start and like for later if they want to watch the recordings, it might be like better. So I will share my screen. So today we're gonna speak about the eukaryotic cell. Eukaryotic cells contain various membrane-bound organelles, including endoplasmic reticulum, Golgi apparatus, mitochondria, and lysosomes. And these organelles collaborate to perform functions essential for the cell's survival and well-being. And they have very high ability to form tissues and organs. I guess these things you already know them. Cytoplasm is a complex gel-like substance that fills the cell and surrounds its organelles. It's made up of water ions, various organic molecules, including enzymes, nucleic acid, amino acids, and sugars. Okay, so you should memorize the composition of cytoplasm for the Imat. This is important. Dynamic nature continuously changes and adapts to meet the cell's needs. The main function of the cytoplasm serves as the medium for chemical reactions within the cell. Cytoplasm of a plant cell contains chloroplasts, which are responsible for photosynthesis and production of energy-rich molecules like glucose. The role of cytoplasm facilitates the movement of materials within the cell, aiding in the transport of organelles, nutrients, and waste products. So as we said, it's a gel-like liquid, so we can say that it's a kind of transport organelle inside or transport part inside the nucleus, inside the cell. The nucleus have something which is this white thing, and this is called the nucleoplasm. Cytoskeleton is a network of protein fibers like microtubules and microfilaments. These are the cytoskeleton, which give the structure and the shape and help in the movement of organelles within the cell. Cytoplasm viscosity. The viscosity of the cytoplasm can vary depending on its chemical composition, molecular concentration, and response to stimuli. So the cytoskeleton, we have the endoplasm and the ectoplasm. And so the central area of the cytoplasm that contains the organelles, here is the endoplasm. However, the ectoplasm, the gel-like outer portion of the cytoplasm, providing support to the cell structure and helping to maintain the cell shape. This is the cell having inside the endoplasm and the ectoplasm, which is here on the outside, so near or so close to the plasma membrane. This is the ectoplasm. Ectoplasm helps in the structure around the cell. However, the endoplasm is the part that have all the organelles. Comparison to a jelly donut, the endoplasm is like the filling, while the ectoplasm is the outer gel-like layer. Cytoskeletons. The organelles in the cytoskeleton, nucleus, endoplasmic reticulum, Golgi, mitochondria, peroxisomes, lysosomes, and vacuoles, all of these are organelles within the cytoskeleton. This is the cytoskeleton. These that give the structure of the inside. Okay. So they have here, as we spoke about them, the microtubules and the intermediate filaments and the microfilaments. We said that the microfilaments are the actin, which are the very thin molecules inside the intermediate filaments, and like keratin, let's say, that gives the structure, if you see them, or keratinocytes, and the microtubules, which are these. And if you can see, one second, if you can see that the rough endoplasmic reticulum, the ribosomes, the mitochondria, all of them are from between this cytoskeleton. The control center of the cell where genetic information is stored, regulated, and the cell functions are determined. This is the nucleus. Encased in a double membrane that acts as a barrier, separating genetic material from the rest of the cell. Genetic material organized into chromosomes, serving as the cell's instruction manual. We have in the nucleus, we have the nucleolus, the nucleoplasm, the nuclear envelope, and the chromatin. So here we're gonna have the nucleus. It will have the inside most layer is the nucleolus. And then we go out, we're gonna have the nuclear envelope. And here we will have the pores, which allow the entrance and the exit of molecules from inside or outside the nucleus. We have the nucleoplasm, which is the cytoplasm of the nucleus is here. And we're gonna have, no, this is the nucleolus, here the nucleoplasm, guys. And here the nucleolus. So like this is the nucleolus. Here nucleoplasm, and then here is the nuclear envelope or the nuclear membrane. And then here we have inside, we have the DNA, and this DNA is called the chromatin or the chromosome. Nucleolus, nucleoplasm, chromatin, pores, nuclear envelope, which is this on the outside, and here we will have the endoplasmic reticulum. The nuclear envelope and the endoplasmic reticulum are stuck to each other. Nucleolus is a non-membrane bound organelle involved in the synthesis of ribosomes. Okay. Nucleoplasm, the gel-like substance that fills the nucleus and surrounds the chromatin. Chromatin contains the cell's genetic material visible under a microscope. Ribosomes, small organelles within the nucleus responsible for protein synthesis. Nucleolus, a non-membrane bound organelle involved in, we just did the same. Okay. Nuclear pores are large structures, these are the pores here. Large structures that facilitate the exchange of materials between the nucleus and the cytoplasm. The location is embedded in the double membrane of the nuclear envelope. Composition comprised of the nuclear pore complex with over 30 different proteins. Function: nucleoporins, Fg domains, create a selectively permeable barrier. Nuclear envelope and lamina, essential for regulating nuclear transport. The nuclear lamina is here. Okay, and you should know about all of this. The nuclear pores and the nuclear lamina is the very most inside of the nucleus, which is after the envelope. And you should know that this lamina and this envelope, these two here, they are essential for the nuclear transport. What does transport means? With the exchange of molecules between the nucleus and the cytoplasm, which is the outside of the cell, which is here. The inside of the cell, but outside of the nucleus. Okay, and the MPC has over 30 different proteins. And okay, one second. I want to ask a question. I guess it came on the Imat or might like it's a nice question to ask. Okay. Perfect. So they might ask you, good morning. Okay. So guys, they might ask you that what is the organelle? Okay. So what are, let me write the question here. Organelle, or the organelles in the cell that have a double membrane. So they are the mitochondria. It has the outer membrane and then the inner membrane, which is called the crista. So the mitochondria has the outer membrane and the crista. We have chloroplast. Okay, they have also an outer membrane and an inner membrane. Inner membrane is highly folded to form thyroid membrane. We have the nuclear envelope that we just studied. Also has a double membrane that surrounds the nucleus. Okay, and we have the peroxisomes. Also has a double membrane. So we have single or double layered membrane. Okay. Bravo, Himera, exactly. Mitochondria, chloroplast, nuclear envelope, and the peroxisome. Okay, now let's go continue the slide. No. Okay. So the chromatin is a DNA protein complex within the nucleus that form the chromosomes. Chromosomes are compact structures of chromatin containing genetic material with a varying number across different species. States of chromatin, we have the euchromatin and the heterochromatin. The euchromatin are loosely packed, allowing for active gene expression. This is so important to be on. And the heterochromatin are highly compacted, typically associated with gene repression. So gene expression, euchromatin. Gene repression, heterochromatin. If you want to memorize them in a way, E, E, R, hetero, R. These are loosely packed, the euchromatin, and these are highly compacted or highly packed, the heterochromatin, and allow the gene expression, the euchromatin. However, the heterochromatin are associated with gene repression. So the chromatin has this, it's super uncondensed. The chromatin looks like this. Okay, so then they start to condense and they start to be more shaped and more compacted, and then they will create the two chromatids or the chromosome. Okay, here the histones. What are the histones? Histones are associating proteins at the end of the chromosome. They help in replication, they help in, in division, they help in everything. Histones help in everything about genetic material or the DNA. So they are associated proteins at the end of chromosome. Histones, proteins around the DNA, is wrapped to form nucleosomes. Euchromatin loosely packed, making it more accessible for gene expression or transcription. Heterochromatin are highly compacted and tightly packed chromatin, and they are less actively transcribed or more repressed, more for gene repression. Histones play a crucial role in structuring and organizing DNA in eukaryotic cells. Modifications can undergo undergo various modifications such as acetylation, methylation, and phosphorylation, which influence gene expression and the chromatin structure. Okay. Okay. Perfect. So they can undergo, these histones can undergo so much changes and so much modifications. It depends if we added the methyl group, methylation, or acetyl group, acetylation, or phosphorylation. And this helps in the influence of gene expression and the structure of this chromatin. Methylation increases the gene. No, let me just make sure about this. Okay. So methylation represses the gene expression and acetylation increases the gene expression. Methylation stops this gene expression, however, acetylation is the complete opposite. Makes it easier. Chromatin structure can shift between a more open euchromatin or closed heterochromatin state depending on the cell's gene expression needs. The dynamic balance between euchromatin and the heterochromatin is essential for proper cellular function and gene regulation. Types of histones, we have H2A, H2B, H3, H4, and they play a very important role in chromatin regulation. Histone modifications, these modifications such as acetylation, methylation, phosphorylation significantly influence gene expression, and they are packed in the three. These are the histone shapes. You see H1 is filamentous, these are spherical, and yeah. H2A and H2B are involved in stabilizing with the nucleosome structure. H3 and H4, they play key roles in regulating gene expression. H1 helps organize and compact the chromatin into order structures. Ribosomes, they are small essential structures found in all living cells. They are tiny protein factors responsible for producing the building blocks of life. Requirements for protein synthesis, ribosomes need amino acids, mRNA, tRNA. These are the tools to undergo translation. Without ribosomes, cells cannot produce the necessary or the crucial proteins for survival. We spoke about this before. We said that the subunit will start jumping and then they will create this polypeptide chain. Ribosome structure differs between eukaryotic and prokaryotic cells. These are for memorizing. Eukaryotic ribosomes are ATS composed of 60S and 40S units. Prokaryotic ribosomes, as noted, there are 70S, consist of 50S and 30S subunits. Okay. The subunits are the large subunit and the small subunit. So the subunit bonding, the two subunits are held together by non-covalent bonds and can dissociate when not actively synthesizing protein. So non-covalent bonded. These two subunits are non-covalently bonded. However, they are hydrogen bonded. Why? Because we have here the mRNA. No, we're gonna have here the mRNA, we have here the tRNA, and then we have between them the nitrogenous bases, and the nitrogenous bases, when they bond together, they form hydrogen bond. Ribosomes are chefs. Ribosomes function like chefs, using the mRNA recipe to create proteins. The translation process, ribosomes read mRNA during translation, adding amino acids in the correct sequence to build proteins. Protein synthesis regulation. The process of protein synthesis is regulated by factors such as hormones, nutrients, and environmental conditions. Eukaryotic cells, ribosome size is ATS 60 and 40. Location, it's in the cytoplasm and on the endoplasmic reticulum, the rough endoplasmic reticulum, and the free ribosomes. Prokaryotic cells is 70S, 50, and 30, located in the cytoplasm. So both of them are located in the cytoplasm. However, in the eukaryotic cells, you might find them on the ectoplasmic reticulum. Prokaryotic cells, you will see them free in the cytoplasm, like this. S in the in ribosomal weight refers to the sedimentation coefficient. Its function indicates how quickly ribosomes sediment in a centrifuge, often used as a size measure for ribosomes and other cellular particles. The process purified ribosomes are suspended in a solution and subjected to ultracentrifugation. Ultracentrifugation, the sample is placed in a tube and spun at high speeds in a centrifuge to separate components based on their sedimentation rate. So the S, when we say 50S or 60S or whatever, this S means the sedimentation coefficient. And just speaks about the velocity of this ribosome to be able to centrifuge, and it's also a kind of expression for the size of this subunit of this ribosome. So sedimentation coefficient, measure of ribosome sedimentation speed in centrifuge, ribosome isolation and purification achieved through centrifugation and ultracentrifugation. Ultracentrifugation involves spinning ribosomes at high speeds for sedimentation. Ribosome structure, composed of a large subunit and small subunit. And the role of ribosomes assemble amino acids into proteins during protein synthesis. The mitochondria, we have the crista, we have the outer membrane, we have the matrix, we have the cytosol, we have the inner membrane. This is called the intermembrane. This is called the inner membrane, which is the crista. We have the ribosomes around. We have the DNA, of course, and if you see it's circular DNA. Okay, so it looks a kind of prokaryotic cell. Okay, we spoke about this. So endosymbiotic theory explains the origin of mitochondria. We said that the mitochondria used to be a prokaryote, used to be called the alpha proteobacteria. Mitochondrial origin, mitochondria are believed to be descendants of ancient bacteria that were engulfed by a host cell. Historical context, this event occurred billions of years ago when Earth was inhabited solely by prokaryotes. Engulfment, a large prokaryotic cell host engulfed a small bacteria. Resistance, the bacterium managed to resist the host's attempts to destroy it. Symbiosis, over time, the host cell recognized the bacteria as a beneficial symbiont, allowing it to live and reproduce within its own membrane or within the organelles it was able to. Okay, so here is the kind of figure explanation of this mitochondria. So we have a aerobic prokaryote, okay, and then it entered this host cell, and then the mitochondrion was it inserted this mitochondria inside this host cell. After it inserted it, okay, when they said photosynthetic prokaryote means they are trying to also insert the chloroplast. So the plastids and which are the chloroplasts, the chromoplasts, and these kind of things. So both of them here, when they were inserted inside this host cell, they were able to adapt with this other organelles around, and they attempted all the cell all over the years. Okay. Yeah. Energy production. Mitochondria produce energy for cells through cellular respiration. They play a vital role in maintaining cell health and function. They found in both plant and animal cells. They generate ATP as the cell's main energy currency, and mitochondria produce energy for cells through respiration. They are oval-shaped, double membrane organelles, contain their own ribosomes and DNA with numerous unfoldings known as crista, the primary site of ATP production, surrounded by the crista and contain enzymes crucial for ATP production, which is the matrix. outer membrane is the porous, allowing the passage of small molecules. Inner membrane selective and regulates the passage of ions and metabolites and establishes an electrochemical gradient for ATP synthesis. Intermembrane functions: site of the electron transport chain, site of citric cycle or Krebs cycle inside. So this inside the inner membrane. What happens in this mitochondria? So here, this mitochondria as any other cell, because all cells they need to respire, and however the mitochondria different than any other cell, it respires and also it creates energy. Like it takes energy and creates energy. So it wants to respire oxygen and like gives CO2 after it's burning. Normal, like in the systematic circulatory system that we have in our body, all the cells need to respire oxygen and need to let out the carbon dioxide. So as any other cell, it needs to respire oxygen. When there is oxygen, when this arteries are giving this mitochondria oxygen, when this oxygen exists, what happens here? The glycolysis will happen, and we have oxygen. That's oxygen. What will happen? This pyruvate that was out of this glycolysis will enter inside this inner membrane or the crista and will go inside this matrix and will make the Krebs cycle. If there was a lack of oxygen in the mitochondria, we won't have the ability of this pyruvate to enter, so it will stay outside in the cytoplasm. And what will happen in the cytoplasm? This glucose, this pyruvate molecule will attend fermentation, not Krebs cycle. After the Krebs cycle comes the electron transport chain. So these two are combined. Fermentation, no oxygen in the mitochondria. And the Krebs cycle electron transport chain, there is oxygen in the mitochondria chain. About these, we'll speak more in details, I guess, today in the second session or maybe tomorrow. Okay. Okay, so here we will have the, okay, this is the Krebs cycle, but we're not yet here. So we're gonna speak about this later, this citric acid cycle, right here. But we didn't reach here, so let's leave it for later. Mitochondrial variation vary number and concentration based on the energy requirements of different cells. Self-replication capable of replicating themselves to meet increased energy demands, and particularly abundant in cells with high energy demands such as muscle and nerve cells and even the sperm, since it needs so much energy to be able to move and to be able to be active to hijack the ova. So as a summary, mitochondria are the powerhouse or energy factories of the cell responsible for producing ATP, the main energy currency of the cell. They convert nutrients such as glucose into ATP through a process that uses oxygen and produces carbon dioxide as waste, which is the glycolysis. Glycolysis is C6, 6, this glucose, plus 6O2, gives 6CO2, plus 6H2. Plus heat or energy or ATP. This is the reaction. The shape is of oval-shaped double membrane organelle. Outer membrane is porous and allows the passage of small molecules. Inner is highly folded. Okay. ATP production occurs primarily in the inner membrane, where the crystal provide a large surface area for the enzymes involved in ATP synthesis. And they mean by this, in the inner membrane, they mean by the citric acid cycle. In addition to ATP production, mitochondria are involved in apoptosis, programmed cell death, the synthesis of certain lipids and hormones, and generation of reactive oxygen species. Okay. And presence in cells, mitochondria vary in number and concentration depending on the cell's energy requirements. For example, they are abundant in high energy demand cells such as muscles and nerve cells. Exceptionally, red blood cells do not contain mitochondria and do not contain nucleus. Peroxisomes, small single membrane bound organelles found in cytoplasm eukaryotic cells, similar in size and shape to lysosomes. Okay, so they said here single membrane bound. They can be both single membrane or right here double membrane. It depends. Okay, they break down biomolecules involved in the catabolism. What does catabolism mean? It's a kind of metabolism that we explained at the beginning of the session. Means decomposition of fatty acids and amino acids. Detoxification, detoxify harmful substances, including alcohol, by converting them into less toxic compounds. Lipid synthesis, they play a role in the synthesis of certain lipids such as plasmalogens, and which are important for cell membrane integrity and function. So lipid synthesis, detoxification, and breaking down of the biomolecules. So peroxisomes, they have a lipid bilayer membrane, which is so normal because all membranes are made up of lipids in a very, very high percentage. Inside you have the crystalloid core, and it's not always present, and that's all. Peroxisomes, they contain enzymes that utilize oxygen to break down biomolecules, including fatty acids and amino acids. Okay, we spoke about these three. Simple structure, peroxisomes have a straightforward design consisting of single membrane that encloses enzymes and other molecules necessary for their function. Unlike lysosomes, which originate from the Golgi apparatus, peroxisomes are formed from the endoplasmic reticulum. So here we have the endoplasmic reticulum, the nucleus, the Golgi apparatus. The plasmic reticulum, they form the vesicles, which are called the peroxisomes. The Golgi apparatus, they form the vesicles, which are called the lysosomes. But both of them have almost the same exact function, which is like killing and phagocytosing and breaking down and this thing, detoxification, which have enzymes that helps them to do this. Here we have a comparison between them. Here we have another comparison. Here we have another comparison. So let's see the comparison. So the function of peroxisomes involved in the breakdown of biomolecules, detoxification, and synthesis of certain lipids. Lysosomes break down of certain biomolecules, primarily those coming from endocytosis and autophagy. Enzyme activity contain enzymes that use oxygen to break down biomolecules such as fatty acids and amino acids. Lysosomes contain a wide variety of enzymes that can break down all types of biomolecules, proteins, lipids, nucleic acids, and polysaccharides. Their origin, they formed from the endoplasmic reticulum, formed from Golgi apparatus. They convert harmful substances into more harmless compounds, the peroxisomes. However, the lysosomes, they break down large complex substances into simpler ones that can be used by the cell. Presence in cells, most is more found in most eukaryotic cells with a crucial role in the liver. Found in all animal cells and are particularly abundant in immune system. Immune system, why? Because lysosomes, they undergo or they make the process of phagocytosis. And peroxisomes, why the liver? Because they make the process of detoxification. Plant cells, they have this cell wall made up of cellulose, and they provide structure and protection. And animal cells, they don't have cells. Okay. So the vacuoles in the plant cells, usually one large central vacuole storing water, nutrients, and waste products. However, the animal cells, they have vacuoles, smaller vacuoles more dispersed throughout the cell. Plastids, they are present, involved in synthesis and storage of pigments like starch, like lipids, and they even give the color, like the color of the fruits, they give the color of the green plants and whatnot. Animal cells, they don't have that. Centrioles, they are absent in the plant cells. Other structures organize spindle fiber during cell division. However, they play a very huge role in the cell division of the animal cells. Centrioles are these two sides of the spindle fiber from up to down. If you see these two sides, these are called centrioles. When the spindle fiber is disappearing, these centrioles sit together, right? They align together next to each other. Listen. Lysosomes, they typically absent in the plant cell. Similar functions are performed by vacuoles and peroxisomes. So usually peroxisomes, they make the same function in the plant cell of lysosomes. However, they are super present in the animal cells and they are responsible for breaking down waste material. Cytoskeleton is present and more expensive and less rigid than an animal cell. However, it's less expensive and more rigid than shape and size. It's more rigid, rectangular or square due to the cell wall, generally larger. However, the animal cells are more flexible, can adopt various shapes, generally smaller. Why? Because its shape is like this. Animal cell does not have a cell wall, that's why. That's why it's small. Chloroplast overview. Specialized organelle found in plant cells from alga. Primary function responsible for photosynthesis, that process, the process that converts light energy into chemical energy. So photosynthesis reaction is the complete opposite reaction of glycolysis. So 6CO2, 6H2O gives C6H12, 6 plus 6O. This is the opposite one of the glycolysis, and this is the reaction. They produce glucose, a sugar used as an energy source for the cell. They contain chlorophyll, the pigment that captures light energy. They are important for sustaining the energy needs of the plant through the synthesis of organic compounds. The chloroplasts are found in the mesophyll tissue of leaves, this for memorizing, where they receive optimal light exposure. Pigments chloroplasts contain pigments like chlorophyll that absorb light energy for photosynthesis. Energy conversion, chloroplast uses light energy to convert water and carbon dioxide into glucose and oxygen through the Calvin cycle. The byproduct, oxygen produced during photosynthesis, is released into the atmosphere. Shape and structure, they are disc-shaped or like a CD shape and surrounded by a double membrane. Internal components inside the chloroplast, there are thylakoid membranes and stroma. Thylakoid membrane are the site of light-independent reaction, and the stroma, they are the light-independent reaction or the Calvin cycle of photosynthesis to occur. So if you see thylakoid, it has an L, means it's light depending. However, the stroma does not have an L, so it's light independent. I memorize it in this way. Outer membrane composed of lipids, it encloses the chloroplast. Inner membrane also lipid base. It surrounds the stroma and contains proteins essential for photosynthesis. Thyroid membrane made up of proteins and pigments like chlorophyll arranged in stacks called grana. These are how they look like. Thyroid. All of them called grana or granum. Another one, let's say, another granum. These two granum together are called granum. The thyroid lumen, the space inside the thyroid membrane, which is this, is called the lumen inside of the thyroid. Okay. Yeah. Chloroplasts, they are specialized organs found in plant cells and alga, essential for the process of photosynthesis. Photosynthesis is the process that transforms light energy into chemical energy, resulting in the production of glucose and oxygen. The location in the mesophore tissue of the plant cells, they capture light energy using pigments such as chlorophyll and utilizes it to convert water and carbon dioxide into glucose. Okay. The structure is disc-shaped organelles with a double membrane. They contain thyroid membranes, site of light-dependent reaction, and the stroma-independent reaction. Outer and inner membrane composed of lipids, but also the outer and the inner both of them composed of lipids. Thyroid membranes with proteins and pigments organized in stacks called grana. Thyroid lumen contain water. Enzymes in the stroma facilitate the synthesis of glucose from carbon dioxide and water during photosynthesis. So the enzymes here have light energy absorbed by pigments in the thyroid is converted into glucose and oxygen. The glucose is used for energy and the oxygen is released into the atmosphere. So then uses the glucose for the energy. Perfect. This is done. Now, because I was trying my best to be fast, to be able to finish the slide in this hour, because I might do next hour chemistry. So let's see the chat. Okay, Said, don't worry. I'm recording, don't worry. Okay. So, okay, 1 second. So I want to speak sometime about the photosynthesis before this hour ends. Okay. So we were saying that the reaction of the photosynthesis is CO2. What kind of reaction is this reaction? Okay, this is the photosynthesis, this is the glycolysis. Let me draw the glycolysis. Let's see the kinds of reactions. So here the glycolysis. It's the complete opposite, C6H12O6 plus 6O2 gives 6CO2 plus H2O plus heat. Here heat or energy plus, okay. Let's see. What kind of reaction is the first one? What kind of reaction is the second? This is in the class. Good morning, Ifra. Good morning, Angela.
Good morning.
Good morning. Bravo. And the second? Bravo. Okay, I want you to give me from the chemical side of the reaction. What kind of reaction? So the first one is indeterminate because it's getting like it's using heat And the second one, it's producing heat. So the first one is it's a redox reaction, the oxidation reduction reaction. The second one, since we have oxygen, we have carbon dioxide, we end up with water and heat. This is a combustion reaction. We are just heating glucose up, Combustion. We are combusting the glucose, let's say, because when we have oxygen as a reactant, carbon dioxide as a product, heat as a product, we are combusting. Okay, the heat here or the energy here, let's say, from where does this photosynthesis is using? From where it's breaking this energy? This reaction, it needs energy. From where is this energy of the photosynthesis? We spoke about it in the slides, so let's see. Glucose, excellent. So we have here a byproduct, oxygen and the glucose. It uses this exact glucose after it does the first photosynthesis. It stores a part of the glucose and then it uses it for the next photosynthesis. So here is the combustion, here is the redox. Why here is the redox? Because we will have the CO2 will be reduced in the glucose, so CO2 is reduced, and H2O will be oxidized into oxygen. That's why it's a redox reaction. Okay. Okay, now let me explain of something. I'll draw a chart. So the chart looks like this. Here we have light reaction, and here we have carbon cycle. Here we have ATP and ADPH. And here we have ADP plus inorganic phosphate and ADP plus. Here in the carbon cycle, it will uses carbon dioxide and it will produce glucose. And then, not just glucose, can produce any kind of carbohydrates, any kind of energy product. And then here in the light reaction, it will use the water. It will split the water. So how the reaction of water will happen here? It will be H2O, O2, H2O gives O2 plus H plus. Okay. So when this division of the water molecule it will be divided, oxygen will be produced. So here oxygen is produced. And here how the light reaction happens. The sun will hit the chlorophyll or the chloroplast and will make this electron on the inside the chlorophyll to excite and to produce a photon. How the excitation happen, I will explain now. So this light reaction happens as we said in the thyroid. Here we have L, light reaction, and this happens in the front or the light independent reaction. Yeah. So as we said, the light reaction. No, number one, here we start the light independent reaction. So it uses carbon dioxide from the air. It undergoes the Calvin cycle. It produces glucose, and then what happens that it uses the ADP and the inorganic phosphate with NADP plus, and then it undergoes the light reaction. It uses water. It splits the water. It produces oxygen using how it splits the water. This photon from the sun, after exciting this electron, this photon of the sun will be able to break down this water molecule and then produce the oxygen. Here happens in the stroma, here happens in the cytochrome, the independent stroma, the dependent light reaction, and the cytochrome. And okay, okay. So okay, perfect. I guess mm, yeah, and this photosynthesis happens in plants plus some bacteria. Okay, what else we should know? We should also know something that we have 20 to 100 chloroplasts in a cell and 0.5 millimeter cube chloroplast in a leaf. These also you should know them. Photosynthesis is an endothermic reaction. Glycolysis is an exothermic reaction. Okay. So for now, that's all. We're gonna speak more in details about photosynthesis for later, for today maybe, or the glycolysis. So this is more for later, but for now, like everything is okay.